let's get into it. Innate immunity. We are going to focus on mammals, but all animals have some kind of innate immunity. For organisms like cnidarians, for example, you can imagine it's a lot simpler than what we're going to talk about for mammals, but we're going to focus on mammals and specifically human innate immunity. There are two types of defensive mechanisms in the innate immune system. The first are barrier defenses. These are non-specific. You could think of them as anatomical barriers. Basically, they're going to prevent things from getting into your body or prevent them from getting away from the parts of the body where we let them enter. Barrier defenses can be either physical or they can be chemical, and you could probably be thinking of some barrier defenses right now. One major example of a barrier defense is your skin, which is part of the integumentary system. That is, I think, the only system in that big table of systems that I showed you like way back earlier in the semester, I think during the homeostasis lecture. The integumentary system, I think, is the only system on that table we haven't covered. But the big idea with the skin is that it's an outer covering and it's not penetrable by bacteria or viruses. And there's a number of reasons for this. One is that it's just a physical boundary. It's cells that are, it's layers of cells that are, together, and stuff can't kind of get in between them, and it also can't get through the plasma membrane of those cells. In addition to just being a physical barrier, the skin is a habitat for commensal bacteria. So you have bacteria that live on the surface of your skin. They live all over your body, just everywhere. You're covered in bacteria. Sorry, but that's the case. And these bacteria are good because, one, they don't hurt you. They just hang out on your skin. Maybe they eat some dead skin cells or something, and they eat other stuff that you come in contact with, but they're not pathogenic to us. And they outcompete bacteria that could be pathogenic to you. They're really well adapted to the surface of our skin, whereas other bacteria that might cause damage to our skin are less well adapted to that environment. So even if potentially pathogenic bacteria get onto our skin, the commensals outcompete them and they don't get a foothold and therefore can't make us sick. In addition to being a physical barrier, the skin also functions as a chemical barrier. So we have oil and sweat glands all around our skin. In some places they're more dense than others, but they're all over the place. And they actually cause the skin to be somewhat acidic. In different parts of your body, the skin pH is going to be between 3 and 5, which is not super hospitable for most organisms. And in particular, since we just heard that like the pH of your blood is a little bit basic, it's like 7.4, if something is going to do well living inside your body, then it's going to have a problem living on that slightly acidic environment on your skin. So it has a tough time getting in. Another barrier defense is the mucus lining of various parts of your body. So mucus is this thick, viscous material that traps pathogens. Think about like mucus in your throat or your nose, right? Boogers are a really good example of mucus that is good at trapping pathogens. So we find mucus in our digestive, urinary, respiratory, and reproductive tracts. Basically, anywhere there's an opening into the body, usually there's mucus or some other substance that the body makes that's going to make it hard for pathogens to get into the body through those openings. In these openings, we also frequently see ciliated epithelial cells. These are cells with cilia that are moving the mucus outward. So they're creating a current where the mucus is moving out of the body. So the bacteria tries to get in, it gets stuck in the mucus, and then the mucus is kind of ejected from the body via that ciliary action. One last component of mucus linings is an enzyme called lysozyme, which is able to destroy some bacterial cell walls. So for some bacteria, they get trapped in the mucus, and then this enzyme just slowly breaks down their cell walls so they can't invade your body. Those are examples of barrier defenses. Another extremely important barrier defense is found in the stomach. So we've talked about the environment in the stomach before during digestion. What's the main thing that we make in our stomach, or one of the main things that we make in our stomach when it's actively digesting? Hydrochloric acid. So hydrochloric acid creates a pH of about 2 in the lumen of the stomach. There's not much that can survive at a pH of 2. So we ingest a lot of microorganisms in our food, but that stuff gets into our stomach and it mostly gets killed by the stomach acid. The stomach is a really important chemical barrier defense in that regard in terms of preventing pathogens from getting further into your body. But in spite of all of these mechanisms, pathogens do get through the barriers. Once a pathogen gets into the body, how do we deal with it then? The answer is cellular innate defenses. These are cells, and mostly we're talking about white blood cells here, but these are cells that destroy pathogens once they get into the body. One major cellular component of the innate immune system are phagocytic cells. And these cells do exactly what they sound like. They ingest pathogens via phagocytosis. So right here, we have, this is a white blood cell, it's a phagocytic cell, and in here in green, we have bacteria. Now this is a scanning electron micrograph. It's artificially colored. It doesn't actually look like this, but just so you can tell apart what's going on here, this is the phagocytic cell, and then these are the bacteria getting eaten via phagocytosis. Now, phagocytic cells are one type of cell that I mentioned earlier that can actually leave the blood vessels, so they can crawl throughout the body. During an infection, we often find phagocytic cells in the lymphatic system. So the way this works is the phagocytic cells, they leave the bloodstream, they get into the interstitial fluid, and ultimately they get into the lymph fluid. And they move through those lymph vessels, gobbling up bacteria. What they'll often do is accumulate in the lymph nodes and basically set up like checkpoints, like a roadblock, where they're saying, okay, and they're kind of checking everybody as they go through, and if they find any bacteria trying to move through the lymphatic system, they can gobble up those bacteria. This is why lymph nodes are often swollen when you have an infection. And if you go to the doctor with symptoms of an infection, the doctor will often check for swollen lymph nodes because the accumulation of phagocytic cells in the lymph nodes often blocks the fluid flow. So you have the lymph nodes swell up as the fluid accumulates because the phagocytic cells have kind of blocked the pathways in and out. Basically, you could think about this like a roadblock causing a traffic jam. So everyone's got to slow down for the toll plaza, either hand over your money or just scan your Easy Pass, and that slows all the traffic down. That causes a traffic jam at the lymph nodes swell up, and your doctor can then see those swollen lymph nodes as an indication of the presence of an infection. As members of the innate immune response, phagocytic cells are not recognizing specific pathogens. Instead, they're just recognizing things that are not you. They're recognizing pathogens based on specific chemical markers that we do not have. They do this by using receptors called toll-like receptors. Now, there's a lot of different types of toll-like receptors. The mechanism is really interesting. We're not going to worry about any of that stuff. All I want you to know about toll-like receptors is that they bind to molecules that are characteristics of pathogens, but absent from vertebrates, and in particular for us, absent from mammals. So I'm going to go through some of these things. You're not going to have to know like the specific receptors, like TLR4, TLR5, doesn't matter. What I just want you to know are some examples of things that bind to toll-like receptors because they are characteristic of viruses or bacteria rather than mammals. Again, the idea here is that when a phagocytic cell detects any one of these substances, it can be sure that that thing is not coming from us. It's coming from a potential pathogen, so you need to destroy the cell that's associated with it. So here are some examples. One type of toll-like receptor detects lipopolysaccharides. Where do we find lipopolysaccharides, or LPS? Yeah, they're the outer layer of the gram-negative bacterial cell wall. We don't make lipopolysaccharides. The only place you find them is gram-negative bacteria. So if we detect lipopolysaccharides, we need to try to kill the lipopolysaccharides. Another example is double-stranded RNA. So we talked about this a little bit when we talked about viruses. In our body, we mostly use single-stranded RNA. Our cells have a little bit of double-stranded RNA, but very, very little. If there's any appreciable amount of double-stranded RNA in a human cell, that's coming from a virus. So if a phagocytic cell detects double-stranded RNA, that means there's a virus around, kill the virus. And then one more that you don't need to know. This part is just me being me. You don't need to know this, but it's one of my favorite things. It's CpG DNA. And all that means is DNA where you have cytosine immediately followed by guanine, and the P represents the phosphate group in the backbone of the DNA. So CpG DNA. What in the world is up with CpG DNA? Well, mammals, for whatever reason, minimize the amount of CpG DNA, or CG dinucleotides, sequences of two bases where it's C followed by G. Mammals minimize that in our genome. We have very little CpG in the human genome. Anytime our cells detect CpG DNA above a certain frequency, that's an indication of a pathogen because we don't have it. Now, this is really interesting because CpG DNA is one of the things we think made the 1918 influenza pandemic so deadly. Again, you don't need to know any of this stuff about the CpG thing, but basically, when our cells detect something like this with a toll-like receptor, it causes a response that we're going to talk about in a few minutes. And that response can have adverse effects. If your immune system overreacts, that can actually make you more sick than the pathogen itself. And this is actually happening right now with COVID-19. The response that I'm talking about is called a cytokine storm, and we're going to see why it's called that in a few minutes, because we're going to see cytokines in just a few minutes. But the idea is that when your immune system overreacts, it's a positive feedback loop, then that causes this cytokine storm, which actually damages your tissues and makes you susceptible to other pathogens. So in the 1918 influenza pandemic, it was a weird spike among people like 20 to 40 years old, people that should have really good, robust immune systems and not really suffer much from the flu. Well, what happened is that particular strain of influenza had a very high concentration of CpG in its genome. This caused our immune systems to absolutely freak out. So the people with the strongest immune systems experienced cytokine storms. People in that 20 to 40 age range experienced cytokine storms. Well, one effect of a cytokine storm in your lungs is fluid accumulates. And when fluid accumulates in your lungs, it becomes a really good environment for bacterial pneumonia. Now, in 1918, we didn't have antibiotics. So if you got bacterial pneumonia following a case of influenza, your immune system was wrecked, and you're probably going to die from that pneumonia. So one of the reasons why that 1918 pandemic was so deadly was the high proportion of CpG in those viral genomes, in those influenza genomes. We're seeing this right now with COVID-19. A lot of the most severe cases of COVID-19, I'm just reading about this morning, and it is the 17th as I record this. It's April 17th as I record this. And I was just reading earlier this morning that doctors in China are reporting cytokine storm syndrome. I think that's the name of the specific set of symptoms. But it's basically the reporting what it looks like are cytokine storms that are making people sick. It's not the virus directly, it's an immune overreaction to it. So these toll-like receptors are really interesting because they detect things that are not us, such as lipopolysaccharides, double-stranded RNA, and what I find really interesting, CpG DNA that's relevant for the 1918 influenza pandemic, and apparently, I don't know if it's specifically CpG, but the result of that in 1918, the cytokine storm, is relevant for what's going on right now. Now remember, you don't need to know any of that stuff about CpG DNA and cytokine storms. That's just for your information and because I think it's interesting. But you need to know toll-like receptors and that they detect things that are characteristic of pathogens that we do not make, and that stimulates the response from the phagocytic cells. Phagocytic cell activity is something you're all familiar with because we've talked about basically every step in this process before. So I'm just going to briefly go through this flowchart. Basically, you have your pathogen detected right here. Let's call that a bacterial cell. We're going to surround that pathogen with pseudopodia, and we're going to ingest it via phagocytosis. Now we have a vacuole containing those pathogens, and that vacuole is basically a food vacuole, and we're going to fuse it with the lysosomes. The lysosomes, of course, have enzymes to break down those cells. Now, we're not going to use this for food, but so far the process is basically exactly the same as what we saw for phagocytosis for food. You fuse the vacuole with the lysosome. Now those enzymes are going to break down whatever was in the vacuole that you ingested. And then in the case of phagocytosis, we're just going to release the debris from the dead pathogens back out into the environment. That's the basic activity for phagocytic cells. Phagocytic cells are not the only way that our body deals with pathogens. We also have cells called natural killer cells, or NK cells. These cells circulate through the body, and they don't directly detect pathogens, but they detect abnormal surface proteins that are indicative of things like a viral infection or cancer. When they detect a cell that has these abnormal surface markers, they're going to interact with that cell and release chemicals that trigger apoptosis, that trigger cell death in that abnormal cell. So they don't directly attack the pathogens, but they're going to prevent the pathogens from spreading to other cells by triggering the destruction of the affected cells. You can think about this like like if you ever watch like a zombie movie or a zombie TV show, there's the group of people like the scrappy band that's trying to survive the zombie apocalypse. And there's always somebody that gets like bitten or scratched and doesn't tell everybody else in the group, but like a little bit later in the movie they start acting a little bit weird, and then everyone's talking and they're like, "Yo, I don't think Bob's okay. You guys, what do you think is wrong with Bob?" And then someone else kind of realizes what's up and they say, "Look, I'm going to take care of Bob." And then they kind of go off, and then like you don't see Bob anymore for the rest of the movie. So the natural killer cell is the guy that went off and took care of Bob, and Bob, in this somewhat tortured analogy, is the virus-infected cell. It's too late to save Bob. Bob is a lost cause. What we need to do now is prevent Bob from spreading the zombie apocalypse to everybody else. So you got to get rid of Bob. That's what the NK cells are good for. They find the infected cells and trigger their destruction so that the viruses or whatever pathogens can't subsequently spread to other cells. In addition to cells, there are also antimicrobial peptides and proteins involved in the innate immune response. One group of proteins are the interferons. These are secreted by cells that are infected by viruses. They don't save that cell itself, but they're basically signals to surrounding cells to say, hey, there are viruses around, get ready. So it's kind of a selfless act that a cell, rather than fighting the virus, it's going to change gears and kind of give up fighting the virus, but it's going to signal to the surrounding cells, hey, I'm infected by a virus and it's coming for you next, so get ready. So those surrounding cells can ramp up defensive measures to prevent the virus from infecting them. And then we have something called the complement system. We're going to get into this a little bit more next time because it's related to the adaptive immune response. But this is a group of 30 or so proteins that circulate in the blood plasma. And they circulate in their inactive form, but they're activated when they bind to surface proteins of microbes. Once they bind to the surface of the microbes, they trigger the lysis of those invading cells. And we're going to talk about the mechanism next time, because in order to work, it requires part of the adaptive immune system. But basically, you have these cells that go around, and when they bind to the surface of a pathogen, they trigger the destruction of that bacterial cell, which is super cool. And we're going to see that mechanism in more detail next time. When all of this happens, when your body's antimicrobial peptides and the cells of your innate immune system respond to the presence of pathogens, you get a local inflammatory response, an inflammatory response. So due to tissue damage, you get heat, redness, edema, which is swelling, and also pain. It hurts. An inflammatory response hurts. That's why anti-inflammatories decrease pain. We're going to look at a flow chart of how this works. It's going to be 3 parts here. In the first part, we have, like in this case, it's a splinter causing a wound in the skin, and you've got bacteria that are then invading due to that injury. You have immune cells, so you've got various types of white blood cells here. Don't worry about what types. They are going to release histamines and cytokines. And also the capillaries are going to dilate because we have an injury, so we want to bring more white blood cells to that area to fight the invaders. The histamines and cytokines lead to a positive feedback loop of immune system activation. So we're recruiting more white blood cells to this site. We are opening up the blood vessels, so we're dilating the blood vessels, which means we're increasing the blood flow and we're getting more liquid to the site. That's where the swelling comes from. And we're recruiting more and more white blood cells, and as long as they continue to detect the pathogens, they're also going to release histamines and cytokines. This is where that cytokine storm I mentioned a few minutes ago comes from. If this happens too much, you get too much swelling, and this liquid could end up going actually into the alveoli of your lungs, and that can cause a problem. So the cytokine storm is when this happens too much. Other interesting note here with these histamines, these are the things you block when you take antihistamines. What you're doing is stopping inflammation. And this is why, for example, antihistamines can stop itching during an allergic reaction. The itching that you feel from like a mosquito bite, for example, is actually the white blood cells crawling around under your skin, and they're bumping into mechanoreceptors in your skin. And you perceive that as itchy. How cool is that? So when you get like a mosquito bite or something that's causing inflammation on your skin, it's itchy because you're actually feeling the white blood cells crawling around. By taking an antihistamine, you're blocking the ability of that positive feedback loop to amplify the signal, and you're minimizing the white blood cells that are crawling around under your skin, and that decreases the itching. Isn't that awesome? I think that's so cool. So after some amount of time of all of these blood cells getting activated and recruited to the site, eventually they're going to mop up, they're going to clean up the bacteria, the wound is going to heal, and the signal, the histamines and the cytokines are going to go away, which means the white blood cells can disperse back into the bloodstream. The fluid will start to kind of decrease, it'll go back to normal, and the blood vessel will go back to its normal size. So that's your local inflammatory response. You can also have a general inflammatory response due to an infection, basically inflammation systemically across the whole body rather than at a specific site. The main symptom of a general inflammatory response is a fever. Now, a fever is not the same thing as hyperthermia. Hyperthermia is when you are too hot, when your body temperature is above the set point temperature. A fever is a change in the set point temperature of the body, which facilitates an immune response. So a fever is actually helpful. You raise the body temperature, and that, one, increases phagocytosis, and it basically helps your white blood cells function. They actually function better at a slightly higher temperature than 37 degrees Celsius. So you bump it up a couple degrees, and your immune response is going to be stronger. And it interferes with the pathogens, because the pathogens are adapted for 37 degrees Celsius. Well, if you bump the temperature up a few degrees, now you're outside of their Goldilocks zone, and they're not going to function as efficiently. So you raise the temperature, it makes it easier for your body to fight the infection, and it makes it harder for the pathogens to fight back. So as a general rule, unless the fever gets so high that it's dangerous, actually, let me start by saying I'm not a medical doctor, I'm not dispensing medical advice, but in general, there have been studies that shows that you basically recover from an infection faster if you allow a fever to occur rather than treating the fever with something like Tylenol, which is going to reduce that fever. Now, obviously, you want to treat the fever because the fever is miserable. Everyone hates having a fever. And as someone with a toddler who has had lots of daycare illnesses over the last year and a half since she started daycare when she was 6 months old, kids are miserable when they have fevers. So you want to give them something to treat the fever. But there's lots of data that show you actually recover faster when you have a fever compared to when you treat the fever. A fever is an important part of an immune response. So that's the idea with a fever. It's miserable, but it does facilitate a faster recovery in general. And if you're wondering why there is a cowbell on this slide, just go on YouTube and Google Saturday Night Live, Christopher Walken, cowbell. You're welcome. So that's all we're going to talk about in terms of the innate immune system. The idea is that it is general. It detects things that are not you, but it cannot distinguish between different specific pathogens. There are two components to the innate immune response. There are barrier components to it, like stomach acid in your skin, and there are cellular components like phagocytic cells and natural killer cells. An innate immune response, specifically inflammation, involves cellular activity, it involves positive feedback regulation, and it involves physiological changes to your body that facilitate that response.